Starting with qPCR. We start this session by comparing quantitative real-time PCR with legacy PCR. We then discuss the detection methods that are available, reverse transcription, which is needed in RNA analysis to convert the RNA to complementary DNA, quality control, and we end up with optimizing the qPCR experiments. PCR, as it was described by its inventor, Kary Mullis, in 1986, is a technique to amplify a target nucleic acid. To a sample suspected to contain the targeted nucleic acid, primers are added. Primers are synthetic oligonucleotides that are complementary to proximal regions in opposite strands of the double-stranded targeted DNA. Also added are nucleotide 3-phosphates and heat-stable DNA polymerase. Most common is the Taq polymerase from Thermus aquaticus. The sample is processed by varying temperature in repetitive steps. First, the sample is heated to about 95 degrees, which causes the DNA strands to separate. This is called melting. Next, the temperature is lowered to some 55 to 65 degrees, allowing the primers to anneal. In the third step, the temperature is set to about 72 degrees, which is optimum for the Taq polymerase to extend the primers by incorporating the nucleotides, creating complementary copies of the template strands. This effectively duplicates the original double-stranded target molecule. The process is repeated over and over many times. After some 20 cycles, the PCR produces 1 million copies of every target molecule that was present initially. For his brilliant conception of PCR, Kary Mullis was awarded the 1993 year's Nobel Prize in Chemistry. PCR was refined by Russell Higuchi, who added a dye whose fluorescence increased upon binding to the DNA formed. In the graph, the y-axis indicates the fluorescence from the bound dye, and the x-axis indicates the number of PCR cycles. The data shown is a 10-fold serial dilution covering 7 logs of concentration. The more concentrated sample, the earlier the fluorescence appears. Comparing the samples after a certain number of PCR cycles, as is done in legacy PCR, for example after 24 cycles as indicated here by the vertical blue line, the signals from the initially second, third, fourth and fifth most concentrated samples are clearly separated, but we cannot distinguish the signals from the 3 most diluted samples because negligible amounts of product have been formed, nor can we distinguish the signals from the 2 most concentrated samples because here the PCR has saturated and essentially the same amounts of products have formed. Clearly, comparing PCR amplified samples after a fixed number of cycles, the dynamic range is limited. If we instead compare the number of cycles required to reach a certain fluorescence threshold level indicated by the green line, which is equivalent to producing a certain amount of PCR product, we readily distinguish the signals from all the samples. This comparison requires we monitor the amount of PCR product produced throughout the reaction, which is exactly what is done in real-time PCR. Hence, in real-time PCR, the dynamic range is virtually unlimited. The amount of product formed in the course of PCR can be monitored using a reporter dye, whose fluorescence increases upon binding to double-stranded DNA. Several dyes are available, the most popular being the asymmetric cyanine dye, Cybergreen 1, originally developed at Molecular Probes. Cybergreen binds in the groove of double-stranded DNA. Upon binding, internal rearrangement of the dye is restrained, which leads to intense fluorescence. An alternative reporter is the probe. Several probe chemistries are available. The most popular is the hydrolysis probe, also known by its brand name, the Tagman probe, originally developed by Ken Levac. While the dye reporter binds to any double-stranded DNA that is formed, probes are sequence specific. The hydrolysis probe is an oligonucleotide modified with a dye in one end and a quencher in the other end. Upon excitation of the dye, the energy is transferred to the quencher and no fluorescence appears. During the PCR, the hydrolysis probe hybridizes to the template strand upstream of the primer. As the primer is elongated, the Taq polymerase exonuclease activity degrades the hydrolysis probe, separating the dye from the quencher, generating intense fluorescence. While the dye reporter is sequence non-specific and can only be used to detect a single target per reaction, probes are amenable for multiplexing, allowing simultaneous detection of multiple targets. The probes are designed to target different amplicons, each probe being labeled with a distinct dye. The fluorescence from the different dyes are detected in separate channels of the qPCR instrument. The probes also distinguish between the amplicons produced from the targeted sequence and any aberrant amplification products. Dye reporters don't. Dyes bind to any double-stranded DNA formed, generating a fluorescent signal. Different PCR products formed in dye-based qPCR can be distinguished by post-PCR melt curve analysis. After completed PCR, the sample is gradually heated while fluorescence is recorded. As the temperature rises, the double-stranded amplicon melts, the dye dissociates, and the fluorescence drops. The amplicon melts over a rather narrow temperature interval that depends on the amplicon length, its GC content, and to some degree also on the sequence order of its base pairs. The thermal stability of the DNA molecule is specified by its melting temperature, Tm, at which temperature 50% of the molecules has strand separated. This is the inflection point of the melt curve and is typically determined by calculating the first derivative of the melting curve, which is displaced here with a negative sign. Tm is the temperature at maximum of the negative derivative. Dye-based assays should be designed to produce little longer amplicons to make them readily distinguishable in melt curve analysis from aberrant PCR products that are formed through interaction between primers only. Self-priming of primers that leads to extension is a highly unlikely event, but it does occur. Once it happens, the extended product is complementary to the primers and is readily extended in subsequent cycles. Notably, primer-dimer amplification is a complication also in probe-based assays because the primer-dimer reaction competes for reagents and may delay amplification of the target, resulting in too high CQ values. Formation of primer-dimer products is suppressed by avoiding secondary structures in the primers and complementarity between the primers, particularly complementarity at the 3' ends. Hot start refers to a group of techniques and approaches that maintain the reaction inactive after the mixing of the components until the mix is heated for activation. Most common hot start technology uses heat-sensitive antibodies to inactivate the polymerase. Reversible chemical inactivation of the polymerase, primers or nucleotides or other options, as well as sequestering of critical reaction components or physical separation. The handling of the sample is also important. Generally, the polymerase and the primers should be kept separate as long as possible, and once mixed, the sample should be kept on ice. Also, if the PCR product shall be processed further, the reaction mix shall be placed cold and the PCR shall be inactivated if possible. A major advantage of the dye reporter is its less complex design and lower cost. Dye-based assays are usually run using conventional 3-step PCR, while assays based on hydrolysis probes use 2-step protocols with combined annealing and extension steps. The loss of design flexibility may in some cases be a disadvantage. In summary, dye-based assays are preferred for expression profiling when many targets are assayed in parallel singleplex reaction and it is not critical to avoid false positive signals. Probe-based assays are preferred for pathogen diagnostics when false positive signals must be avoided, multiplexing is required, or when closed tube systems are needed. This slide shows some of the first qPCR instruments that appeared on the market, including the famous ABI 7700, also known as the TACMAN instrument, and the original Idaho Technology light cycler. These instruments opened a new era in molecular diagnostics, but they are outdated today. Today's instruments are much more user-friendly and typically smaller. There are impressive dedicated instruments such as the Cobas TACMAN from Roche for clinical diagnostics and the Quan Studio from Life Technologies for high throughput expression profiling and digital PCR. But most labs settle for any of the 96 or 384 well plate instruments that are open and flexible platforms. These are available from a large number of suppliers. When designing assays for transcripts, one shall be aware that primers may also amplify genomic DNA. If the gene has introns, primers can be designed to span an intron or to cross exon exon boundaries. Such primers will not amplify the normal genomic copy of the gene. However, not all genes have introns. Also, many genes, in humans some 15%, have pseudogenes that often lack the introns of the functional gene. Amplification of those pseudogenes cannot be avoided by primer design. Some genes have hundreds of pseudogenes, leading to substantial background from genomic DNA. The background can be measured with RT- controls. In an RT-PCR control reaction, the same analysis is performed, but the reverse transcriptase is omitted in the RT step. The CQ of the RT- reaction shall be at least 5 cycles above the CQ of the regular RT-qPCR for the genomic background to be considered negligible. So here are the RT positive signals and here are the RT negative. This is without treatment with nuclease. And if the difference is insufficient, the sample can be treated with double-strand specific nuclease, which will increase the difference between the RT positive and the RT negative control. Still, an alternative is to use the recently developed valid prime protocol to measure and subtract the genomic DNA background signal. When setting up qPCR in a laboratory, it is advisable to physically separate spaces for pre-PCR work, template addition, and post-PCR to minimize contamination risk. In our laboratory, a person analyzing PCR products in the post-PCR space is not allowed back to the pre-PCR area that particular day. A UV hood is suitable for preparing master mixes. The UV shall be on when the work is not performed in the hood to destroy any nucleic acids, and turned off when working in the hood to avoid burns. When opening a regular test tube, aerosols may escape, contaminating the entire laboratory with PCR product. To minimize contamination risk, use filter tips and screw cap tubes for high concentration DNA samples. Remember, larger volumes are pipetted more accurately. Try to avoid pipetting volumes smaller than 1 microliter if possible. To analyze RNA with qPCR, the RNA must first be converted to cDNA. This is done using a reverse transcriptase, which is an enzyme that transcribes RNA into complementary DNA. The discovery of reverse transcriptases by Howard Temin and David Baltimore was awarded in 1975 the Nobel Prize in Physiology and Medicine. Reverse transcriptases are not very picky about priming, and reverse transcription does proceed even without addition of primer. But it helps adding primers. There are 3 priming strategies. Gene-specific primers can be used to direct transcription to a particular gene. The same oligomer can then be used also as reverse primer in the downstream PCR. gene-specific primers are preferred for diagnostic applications with a single target. Oligo-DT primers can be used to reverse transcribe RNAs with A tails. Most eukaryotic RNAs have A tail. Advantages are that mRNAs are enriched in the cDNA population and that priming bias is minimized. Disadvantage is that the reverse transcription may not reach the region targeted by the qPCR if the RNA is fragmented. An alternative is to use random sequence primers. Random sequence primers hybridize to all accessible regions, effectively reverse transcribing all RNAs, including fragmented RNAs and structural RNAs. Random sequence primers are often hexamers but can be longer. Longer primers are preferred when reverse transcription is performed at elevated temperature. Many kits use a mixture of oligo-DT and random sequence primers for priming reverse transcription. Those priming strategies are preferred for expression profiling when multiple parallel singleplex reactions are performed. Comparing the 3 RT priming strategies on 5 targets, we find that none is superior. Some genes are reverse transcribed at higher yield with random sequence primers, while others show higher yield with oligo-T primers. The difference between the priming strategies can be a few cycles. Critical for efficient RT priming is that the targeted sequence is available. Increasing the temperature may improve hybridization and thus priming efficiency. RNA secondary structures can be predicted using the online tool mFold. The RT yield may depend on the reverse transcriptase used. Here, the serotonin receptor gene transcript was reverse transcribed with 8 different reverse transcriptases, and a yield variation close to 100-fold was observed. This effect is gene dependent. With some genes, RT yield can be boosted manyfold by choosing a different reverse transcriptase, while with other genes there is virtually no effect. RT-qPCR can be performed as a one-step reaction. This is very convenient, but has several drawbacks. Random sequence primers cannot be used. Only a single target can be analyzed per reaction, unless qPCR is multiplexed, and the overall performance is generally lower. Main advantage is that closed tube analysis is possible, making one-step RT-qPCR protocols preferred for analysis of contagious samples. Regular two-step RT and qPCR performed with optimum conditions used for each reaction, and a washing step in between is preferred for most other applications. At each step of the analytical process, a sample can be divided into aliquots that are analyzed in parallel separate reactions. Comparison of the aliquots' CQ values reflect the reproducibility of the downstream reactions. In the left panel, a sample was split into aliquots for qPCR and in the right panel a sample was split into aliquots for RT. The inserts show enlargement of the qPCR response curves in log scale. The substantially higher reproducibility of the qPCR aliquots in the left panel compared to those of the RT aliquots reflects the much higher reproducibility of the qPCR reaction compared to the RT reaction. A good strategy is to perform a fully nested pilot experiment before any important study, starting with a small number of subjects, collecting multiple samples from each subject, and then splitting each sample into aliquots after each important processing step. It is possible to estimate the variance contributions of the different steps. This identifies steps that shall be improved or further optimized, and also help designing and cost optimizing the subsequent larger study. A 4-level nested study with 3 aliquots taken at each level requires 3 by 3 by 3 by 3, which is 81 measurements. This readily fits in a single run. These graphs show examples of nested pilot studies with 4 levels. Subjects, which here were HIFers, sampling, reverse transcription, and qPCR. The left panel shows liver samples and the right panel show blood samples. 4 genes were analyzed in each pilot study. For the liver samples, we see that most of the variation is introduced by the sampling step, reflecting the heterogeneous nature of the solid tissue. For blood samples, substantial inter-subject variation is observed for the cytokines, suggesting that perhaps one of the animals may have been sick. Variation in the sampling step is negligible, as expected for homogeneous blood. In conclusion, probes are preferred for pathogen diagnostics when multiplexing is required and for one-step RT-qPCR protocols. Dyes are suitable for expression profiling, particularly at screening stage when many markers are tested. Be aware that many assays designed to amplify transcripts also amplify genomic DNA. If this is a problem, remove the genomic DNA using double-strand specific nuclease. For reverse transcription, use random sequence primers and/or oligo T primers for expression profiling and gene-specific primers for one-step RT-qPCR protocols. Before starting a large study, perform a fully nested pilot to identify steps that contribute with most variation. Consider means to improve the performance of those steps. Use the result from the pilot study to design and cost optimize the larger study. Be aware that RT-qPCR yields vary among genes and depend on experimental conditions. Always analyze all samples with the same protocol, reagents, and under the same experimental conditions. CQ values are instrument dependent and not readily comparable. Delta CQ values are comparable between samples that have been analyzed using the same protocol. Delta-delta CQ values are comparable even when different protocols have been used.